Go ahead and get started.
Well, good morning, everybody. Thank you for joining us today to this seminar. So today's seminar is part of the Botany and Plant Pathology seminar series during this fall. And today it is my pleasure to introduce Dr. Da-Young Lee. She's a postdoctoral researcher in my lab. Dr. Lee got her PhD in plant pathology from the Ohio State University. And if I'm not wrong, she got awarded the Monsanto Beecher Borlaug Scholarship to get that PhD. Well done, Da-Young. Right after her PhD, she continued to work with Dr. Wang at his research lab, and she became a research associate. And I'm fortunate because she reached out to me one day and she asked questions about possibilities to join my lab here at Purdue. And we're grateful for that opportunity, Dr. Lee. So her current research project involves the use of sensor technologies to describe epidemics of tar spot of corn. However, she is trying to expand those ideas to other diseases. And with that, please, let's welcome Dr. Da-Young Lee.
Thank you for that kind introduction, Dr. Cruz. Thank you, everyone. It's a rainy day for your effort to actually come here and attend the seminar. I would also like to thank those who are joining us through Zoom. Thank you. As introduced, I'm Da-Young Lee, and since me joining the Cruz Lab in September of 2019, I've been here since, and interestingly, it coincided with more or less COVID. And so this project is like my COVID baby. Like it was a product of the time that I was here, but it was COVID. So I'm happy that we were able to be productive. And so, I'll start now. Basically, before me joining this lab, I have been immersed in rice disease related research. I've been working with rice leaf blight, rice leaf strike, rice blast, rice sheath blight, and now I'm working on tar spot. So some of you might be interested, why did I suddenly shift my pathosystem, in which I've been working on rice diseases for nearly a decade? And that's because of the topic that I wanted to share with you this morning, image-based detection and quantification of disease. So as for my outline, I will briefly do an introduction and definition of terms, just for us to be on the same page. And I would move towards sharing my motivations and inspirations that I got during my PhD. That would be motivations, could also be troubleshooting as well. So I would like to share some of that projects that motivated me to work on image-based detection of diseases. And then we move towards tar spot of corn, which is the main character disease of my research. And I will share some of the projects that I've been working on for the past 2 years, and also the collaborations that we had during those times. And I will wrap up my talk with some of the ongoing projects that I have right now and note some future directions. So phytopathometry, it may be a familiar term to some of you. Some may have not heard this term before. At least for me, before 2 years ago, I did not encounter this word. But basically, it just means measurement science in the field of plant pathology. So by definition, phytopathometry is the branch of plant pathology concerned with the estimation or measurement of the amount of plant disease expressed by symptoms of disease or signs of pathogens on a single or group of specimens. So I'm pretty sure that all of us more or less would agree how important it is in our field, plant pathology, or in any science field measurement is. And in the context of plant pathology, phytopathometry is important because it allows us to evaluate the effect of treatments, assess crop germplasm for disease resistance, predicting yield loss, monitoring epidemics, establish thresholds for decision-making, and improve our understanding of disease and so forth. So I cannot emphasize enough how accurate measurements or estimates of disease intensity is important. It directly influences the downstream processes of our experiments and interpretation of results. Faulty conclusions or interpretations would lead us to incorrect actions, and that would in turn affect grower finances as well as the research plan that we have set up. Yeah, we would have to spend more resources. We have to spend additional time. So from the beginning, it is important that we make sure that the measurement scale that we're using to actually use to test our treatments or interpret our results need to be accurate and on point. One of the things that I would emphasize or deeply talk about today is visual estimations, plant disease visual estimations, which more or less all of us are doing right now. So, however, despite the advances of plant pathology from the visual assessments, it is also true that as we move towards precision agriculture, we need more precise and accurate measurements. And one of the limitations of human rater assessments is due to our innate character being subjective. So subjective could mean different things in different contexts. Some people might prefer providing disease ratings in intervals of 10s or 5s rather than giving 1 or a 3 or a 7. There might also be color blindness depending on the person's physical stature. It is also time and labor intensive, so this definitely affects the rater being able to provide accurate measurements, all the samples that he or she needs to evaluate. And of course, if you're doing visual estimations in the field, the heat, the sun, the wind, and the plant canopy would definitely interfere with this. And just to note a few more points, specific points. In terms of counting lesion number, it is reported that lesion number actually affects the rater's ability to provide accurate measurements. For instance, there is a widespread tendency for raters to overestimate disease severity at low severities, less than 10% meaning the disease symptom or the pathogen sign. And inter-rater and inter-rater reliability can be variable, particularly if the person who is rating has not been exposed to training or rating aids. So the more the training, the more reliable the rater will be. But still, that subjectivity point will still be there. So one of the solutions that we wanted to use in order to compensate this limitation that we all have is image-based detection and quantification. So assessment of plant disease severity could basically be categorized into two. One would be human rating, one would be tool or software-assisted analysis of disease symptoms or signs. And so both are considered remote sensing because remote sensing would be defined as something that we do that is beyond direct contact. So visual and imaging would be both remote sensing. So new technologies that offer opportunity to assess disease with greater objectivity. In our case, we used RGB images, at least for me, I used RGB images to do that. And the use of visible light photography and digital image analysis has been increasingly used over the last 30 years. And all the more right now because the cost of camera is just, has significantly dropped compared to 30 years ago. So it allows, it's a more user-friendly method. And so before going into the next chapter, I just wanted to note some of the terms that I'll be using. Plant disease assessments would be to quantitatively measure the amount of disease in the host population. Disease estimates would be the assessments conducted by human raters. Measurements would be the measurements provided by the tool or new approach. And remote sensing would be obtaining information about an object without direct physical contact, which encompasses both visual and tools. Disease intensity would be expressed as either incidence or severity. In our context, we would say that intensity would encompass stomata counts or lesion counts plus severity, and wherein severity is defined as percentage of relevant host tissue covered by symptoms or pathogen signs of the disease. And finally, validation would be comparing the measurement obtained from the analysis, the image analysis, to an actual value or reference data, which would be the human radar assistance for the estimates. So I had two big categories of projects during my PhD. One was rice blast, one was rice sheath blight. I had one of the main projects that I had for rice blast was to generate a novel rice blast resistance using CRISPR-Cas9 by targeting the polymorphic regions shared by PI2 and PIZT, which are broad spectrum origins of rice blast. Aside from that, I had a resistance screening project for rice blast as well. And this is that project. And so with the increase of demand of rice in Africa, with the change of staple food consumption, Africans started to consume more rice, but then they're also relying to exports, meaning the production is not fully supported within the country. So what we want to do is to identify sources of resistance from worldwide rice stocks, particular to the rice varieties, especially bred to thrive already in African growing countries. And so we determined the resistance spectrum of 14 African rice cultivars and 10 USDA rice accessions reported to be highly resistant to East African rice blast isolates. Challenges for rice blast resistance breeding is because of some of the reasons that I've listed here, like inefficient field screening, but majorly driven by the tendency of the rice blast resistance to be broken within 2 or 3 years in the field. So we need to have accurate phenotyping of rice blast resistance, but it also needs to be fast. And so I did the screening of 27 rice cultivars and with a minimum of 3 to 5 replications. And I used this scale, which was frequently used in rice blast disease resistance screening, phenotyping. And it's either 0 to 5, you use a scale from 0 to 5, or you use a scale from 0 to 9. At first, it was easier for me to use the 0 to 5 scale. Because as you can see, if you use the 0 to 9 scale, it just spreads out. And sometimes I would ask myself when I was in my early screening days, should I rate this 2 or 3? It's easier for me to rate 0, 3, 6, and 8, but is it 4 or 5 or 2 or 3? And I hoped that there would be a method where I could do this in a more objective way and in that I could process high number of samples. But I didn't really do anything about it, but I already had this idea that that would be great if there's something like that. And then moving on to another project, I did the rice sheath blight resistance screening project, which is one of the supporting projects that I had for the big rice sheath project umbrella. And so the major project I had was to compare the genome contents of Rhizoctonia solani, pathogen of rice sheath blight, and to see their genomic contents, which drive their tendency to produce, to generate stem lodging, which is the hallmark of rice sheath blight. Alongside that, with that, I also had another rice screening project for rice sheath blight. And that was using Rice Diversity Panel 1, RDP1, which is a collection of purified homozygous Rhizosativa L accessions encompassing land races and elite rice cultivars. So in the end, we were able to identify potential sources of rice sheath blight resistance from RDP1, but we're also able to develop a tool which could accommodate that, which we could use RGB images to actually quantify right sheaf light. And so just a little bit about that process. To determine the potential candidate sheaf light resistance germplasms from RDP1, we did the following screening wherein I would germinate the seeds, put it into pots, grow the Rhizoctonia hyphae, and then we inoculate the actively growing region of the hyphae in uniform amounts, and then place them in at the base of the plant at eggshell and at eggshell. Do the microchamber method using the Petri bottles to enhance humidity within that region, and then phenotype it after 6 or 7 days. Now, during that time, there were a lot of methods to rate right sheaf blight severity, as I've placed a few of them here. But then primarily, my goal was not to grow the whole plant until the end and then phenotype the whole thing. But then what we really wanted was to do screening as fast as possible, as young as possible, but not too young that the plant will just be overwhelmed with inoculum. And one of the ratings that I could have used was lesion height. So lesion height we define as the average vertical height from the base of the plant to the uppermost of the lesion. And then the total plant height would be the height of the base plant to the tallest leaf. However, I faced with several challenges. First, there are no completely resistant rice land races. At that time, we had Jasmine 85, which was known to be moderately resistant. But then with the microchamber method being so effective, it actually produced a lot of lesions. So it wasn't anymore about a total resistance versus susceptible. It needed to be evaluated in a relative sense. As you can see, the moderately resistant cultivar Jasmine 85 still produce these types of lesions, while the lemon was like zombified, but still. And rice sheaf blight resistance is a polygenic resistance. So the varying levels of susceptibility profiles among the different rice cultivars needed a more defined, finite way of measuring the lesions and the extent of disease. And so I asked, is lesion height enough? For visual base scoring, it would be exposed to my rate of subjectivity. For a given plant, for different cultivars or accessions, their heights are not the same. Their column length are not all the same. So given this type of image or this type of sample, if you have this type of lesion, would you still consider the lesion height for this one and say it's susceptible? Or would you say that if there's a bigger lesion at the bottom of the stem, but since its lesion height is small, would you consider that as resistant? So I started asking these questions. And one of the hallmarks of rice sheaf blight was it produced water-soaked lesions. So more or less, it was not like a sporadic here and there, like wherever the inoculum landed, it's just going to, I mean, at least for my experiment setup, we made sure that the inoculum would always start at the base of the plant so that we know the disease progression would be towards the upper canopy. So with the collaboration with Dr. Dong Yap, we were able to come up with a pixel color and coordinate-based k-means clustering. Using the color KMC, it would use the 3 dimensions, the RGB space, and kind of catch the data points which are close to each other will be clustered to each other based on the similarity of RGB values. If we provide another type of information, the pixel row and column data set, it's gonna allow us to consider both color similarity and spatial proximity. And as I've mentioned, the RGB values in the pixels would provide data for color similarity, and the physical location of the RGB pixel would provide a spatial proximity. So our assumption is that the nearer the similar color of pixels from a given point, the likeliness of that being a disease lesion, rather than a very far, but then similar color, because this similar color could occur in different ways. It could be that it's a bit, because with the microchamber method, if the leaves or some part of the plant touches the PET bottle, it kind of rots. And so some regions can be considered as, those are like false negatives. So just to reinforce that color-based statement means clustering, we would provide the information of the RGB image, and then based on closeness of data points, the PCC-KMC would search for the best position of centroids of a given number of clusters with all the 5-dimensional data. And this is a simplified version of color-based KMC, which is the traditional KMC using RGB images. You can see that PCC-KMC was able to cluster lesions that are more proximal to each other. And we were also able to differentiate the region of the lesion as well as those lesions that did not have the lesion. And this was helpful because when we were going to phenotype a specific sample, not only can we have the lesion height now, but also the area of the lesion. And so we would have a higher chance of finding reliable, robust sources of sheath fight resistance using these two types of data, rather than relying only on lesion height. So white tower spot of corn. As we know, United States is the largest corn producer and exporter. And I just included this slide to show how vast corn is being produced throughout the states, especially half of the Midwest and a lot of the states in the East. And this is important because tar spot of corn was first detected in northern Indiana and Illinois in 2015. And the epidemic led to 840 million in losses in approximation. And the stromata that is the hallmark of tar spot of corn is the basis of tar spot detection and surveillance in the field. And as of now, tar spot has continued its way, spreading to different states, even far up to Florida. And recently, Pennsylvania sadly joined the club of the states where tar spot can be found. So tar spot stromata usually are observed during vegetative and reproductive stages. And this is the growth stage, just to show you, for those who are not familiar with corn. And so, but however, we usually see them around VT to the R stages as well. So I've been racing through time right now, but it just as an icebreaker, I would like to try an activity with you guys as well as those who are joining us through Zoom. Let's say that we are all a tar spot reader. The aim of this exercise is not to come up with an accurate measurement, but just to have a feel of how different processes through your vision and your mind is incorporated in you actually providing a disease estimate in a given amount of time. So I could probably allot approximately 10 seconds for everyone. Given that the definition of tar spot would be the percentage of leaf covered with tar spot stromata, could you please give the rating of the sample A and sample B based on that definition, and you can just hold it in your minds. 10 seconds. So basically the area of the black spots relative to the area of the leaf would be the split. So narrowing down your choices, do you think it's going to be 2%, 3%, 5% for sample A, or a little bit more on the right? The tar spot readers are laughing. Based from the tool that we have developed, The answer is sample A is 1.25, approximately 1.25%, and sample B is 7.76. So again, the aim is not, don't be discouraged. I would also be, I didn't get this correct when I did it. So, but now you kind of feel how complex it is to actually come up with the readings, right, in a given amount of time. And if you're like doing field work already, 4 hours in the field, and then you still have to make accurate estimates, that's also a very challenging task. So to help us, we generated the stroma to contour detection algorithm. So before going into that, I just wanted to discuss the data sets that we collected. There's going to be data set A and data set B. Data set A was collected at the University of Illinois Urbana-Champaign South Form in 2019. We collected rice leaves, brought it to the lab, and took images under lab controlled lighting conditions. And the images look like this. So our preliminary approach was how to detect tar spot stromata. But then the idea arose from here. Topographic maps allow visualization of three-dimensional terrain from a flat piece of paper, maps. The feature that makes this possible are contour lines. And these contour lines describe the terrain, and these lines connect the points that share the same elevation. We apply this idea to detect stromata. And to do a bit of a fast forward, the contour lines that describe the stromata is based on the connection of points that share similar grayscale values. So more on that coming up. So the pipeline in detecting tar spot stromata, we used 6 representative RGB images and partitioned it into more than 1,000 image blocks. Now, the type of reference data would be important because that would be our reference to see whether the algorithm is working or is accurate. So first, we provide the input RGB image, remove the background, tile the image, and for every tile, we do a pre-processing of the RGB image so that our algorithm would be more accurate rather than being disturbed with other noises. And then perform the contour analysis, detect the sets of contour lines, and that satisfy our conditions. And then the output of the algorithm would be the number of stromata as well as the area of stromata relative to the leaf area or the sample area. So just a brief, I'm just going to do a brief go through of the process. The RGB image would be removed from its background. We would do pre-processing techniques in order to come up with an image that has no background but only has the region of our interest. And that will be also through homogenization, in which homogenization allows us to adjust the brightness of the image. And we did also a Gaussian filter in order to reduce the resolution of the image. And eventually we would convert that into grayscale images. And this would be the output of partitioning the image, partitioning the whole leaf image into image blocks. So basically, if this is divided into 200 image blocks, it's not one leaf anymore, it's 200 samples. So, and so this is a sample of stromata contour. This would be an image block. And if we zoom out this region, you would be able to see some conglomeration of contour lines. And if we zoom further out, that would consist of contour lines, bigger area contour lines having dense populated contour lines going to the middle. And since it is a grayscale image, the ranges of the value is from 0 to 1. So depending on how much we divide that range, that would also determine the number of contour lines that could be generated in an image. And so for us to validate our method, human rater A, Andres Cruz, and human rater B, Brendan Lane, with the assist of Fiji ImageJ software, we were able to make reference data. So given a image plot, they would use this tool in order to click on the stromata that they believe is true stromata. That was very beneficial, first because they would not be able to click the same stromata twice. Second, that would also provide the location of the stromata within the image that would allow us to corroborate with the region where our algorithm detects the stromata. So that would be that was a very, very good reference data. So we tested the agreement between the human raters as well as the raters and the SCDA algorithm. And to measure the agreement of stromata counts for all image blocks analyzed by 2 independent raters, as well as those of the raters and the SCDA, we used the stromata counts. We collected the stromata counts, and then each image blocks, the number of stromata would be categorized into class 1 to class 5. This is the class that we set out. And we use the weighted Cohen's kappa coefficient, which uses predefined weights that measure the degree of agreement between the raters. And as you can see, the agreement between the human rater A and B was the kappa value was 0.94. Human rater A and SCDA approximately 0.83 and 0.83. So that would correspond to nearly perfect agreement. And this agreement chart further supports that. Another metric to assess performance of our approach is what I've mentioned earlier, which would be the location of the stromata. So here we are going to use precision recall in dyes in order to describe the performance of SCDA with the definition that true positive would be the true tar spots detected by human radar. False positive would be incorrect tar spots detected by the SCDA, and false negative would be the true tar spots undetected by SCDA. And what we would want for this algorithm to show us is that it's producing higher true positive cases and lower false positive and false negative cases, meaning higher precision, recall, and dyes. Here, A, B, C, D, E, F would be the 6 representative images that we got from dataset A. Each leaf would comprise of hundreds of image blocks. These panels would be human rater A. These blocks would be human rater B. We see a general trend that the peaks are towards the right, skewed towards the right, within the range of 80 to 100, meaning that most of the image blocks generated by both raters were in high levels of precision, recall, and accuracy upon comparison with the SCDA, which is good, really good. So more or less, we would also be able to show these types of images where in the red highlighted regions would be the stromata, and this would be the original image. So I hope you guys could also see it. So moving on, we have data set B. We collected this data set in 2020 at the Purdue Agricultural Center at Wanata. We collected 6 time points, August 25, September 3, 10, 15, 22, and 29, and collected a total of 466 RGB images. Remember, the dataset A was all images collected indoors. Now we're going to the field, taking images at broad daylight. RGB images acquired under natural sunlight was done using the Canon EOS camera, the center here. It's the same camera that we used for the indoor capturing of RGB image. And this time, the type of reference data would be the percentage of stromata area relative to the whole leaf area estimated by human rater Mariela Fernandez. And these would be the type of images that. So in collaboration with Dr. Darcy Telenko, this was one of the trial plots that was established in 2020. And this trial was to test fungicide treatments in managing tar spot. And in our previous experience, we already knew that headline AMP was significant in managing tar spots. So we decided to use this as the experimental setup and pick the plots which treated headline AMP as well as those were not treated at all. Fungicide treatments were randomly assigned into blocks and 4 replications were established. 2 plots for replication were selected, untreated and treated, and each plotted consisted of 4 rows, and we would use the middle 2 rows. I would walk in the middle of the second and third row, tag plants, 5 plants in a zigzag manner, and for each plant, I would tag 2 leaves near the middle canopy with the ear leaf in the center. So this would be the disease progress curve generated based on the human disease ratings that Mariela, the human rater, provided. And as you can see, this shows that the treatments significantly is different from each other. This 106, 204, 309, and 406 would be the headline plots, and the rest would be the untreated plots. And for our comparison, we used the middle canopy to compare it with the performance of SCBA. Using the stromata count and percent stromata area, we were able to find that the separation of the severities of the untreated plots and the fungicide treated plots with the use of stromata count and stromata area. We also had a collaboration with Sriram Baireddy, which is a PhD student at the DUP lab, and he generated a pipeline wherein mask region-based convolutional neural network was used. And so the input image would eventually extract the region proposals, do the CNN feature extraction, classify the regions, and detect the objects. So we recommended sliding windows in order to improve his approach, and eventually using his approach, we compared SCBA, mask RCNN versus human rating. And so this will be our final assessment of the SCBA. We have been continuously assessing SCBA for its performance and accuracy. This is the final piece of it. And so here we use lens concordance correlation coefficient, which has been widely used in various disciplines. And then here we use a UDPC of the visual severity provided by Mariela, and then compared it with the stromata counts and area provided by RCNN versus SCBA. And as you can see, we have a much higher PC value compared to the mask RCNN. And I believe that this was a good preliminary data because we have now ideas on how to move forward using machine learning approaches. And this would definitely help us, allowing us to automate our process, our current algorithm, the SCBA process moving forward. And so this is another slide showing further supporting that. This would be human severity, visual severity provided by humans. This would be the disease progress curve using stromata area and counts, like mask RCNN and the disease area. So we can see a similarity here. So as a summary, we had 2 data sets. Data set A, acquired in lab controlled environment, 1 in the field. We generated a stromata contour detection algorithm using this data applied to a more variable type of RGB images, which were collected under natural conditions. We compared human visual rating, SCBA, as well as a machine learning approach in order to come up with the stromata counts and leaf area, at least for these 2 approaches. And then did statistical analysis to test accuracy and agreement. And that also allowed tracking of tar spot progression through time. So with that, we were able to provide stromata counts and area for all the 466 images that we collected in 2020. This work has been published in Frontiers of Science, and it has been it's published. It was recently published last month. And so with that, we also further were able to track the increase of disease severity through 6 time points, which allowed us to do temporal analysis. We were also able to describe the pattern of tar spot progress, and we're also able to select the best tar spot management. It's not really best because we only had one experiment set up, but we were able to decipher the untreated versus untreated, the treated versus untreated. So some of the ongoing projects that we are currently doing right now, using that time lapse, I wanted to focus on the T5 leaves. This is a large version of the T5, the leaf taken at T5 time. What we're trying to do is to see the proliferation of stromata from T1 to T5, and whether we can see first how large does the stromata become from T1 to T5, and to see the relatively recently developed stromata versus those that were developed earlier in the season. And we're trying to work on the calibration right now. And so this is without calibration, this is with calibration. You can see that the different tar spots kind of merged and centered to each other, but then I think we still have some work to do here before moving forward in doing spatial analysis. True positive ratio is also very important. That means that what the human rater sees as true stromata should also be detected by the algorithm. Not only, the counts are not the only things that are important, but then detecting the same thing as a human does is also important. And so I did an annotation of 12 corn leaves from the dataset B. That 12 corn leaves would correspond to approximately 2,400 plus image plots. And we were able to see that the average true positive ratio for all those image blocks were in the range of 70 to approximately 95, which is a good value. And so the annotations would be done like this. We would have to manually delineate the margins of the stromata. And before using stromata counts, we were able to see the precision recall dice values more or less like 50 to 100. Now it has more grouped each other towards the range along the lines of 75 to nearly 100. So using stromata area is indeed has indeed improved the performance of SCBA. We also looked into the type of images, the image blocks that we analyzed. How much disease did we actually get then?
Hey, Christian, can you hear me? It looks like we lost the feed.
Yeah, I don't hear anything either, Damon.
Yeah. Okay, here, I'm going to make you Christian's a co-host. So Christian, you should be able to host now. Amy's computer crashed.
The reason why we're interested in smaller hotspots is
Good.
that we would like to detect it as early as possible. So not only for quantification purposes, it doesn't really have to be. I think those are two different objectives. Quantification is we need to have an accurate measure of the stromata present in the leaf sample. But then this detection thing is more on the early, how small can you detect? How correct stromata can you detect from an artifact or an insect rat? So forth. So that would also allow us, actually push us to generate a large scale ground truth set using the SCDA. So the SCDA would provide the ground truth, and then the mask of RCNN is the algorithm that we would like to further. And we currently also have ongoing collaborations, which are great. We're actually trying to implement SCDA in actual routine experiments, and with Dr. Raksha Singh and Dr. Stephen Lubin, we're working on their NAM lines, trying to phenotype hotspot diseases in their samples. So with that, I would like to acknowledge the people who have been actively working in these projects, starting from Dr. Dongyuk Na to Ram, Edward Delp, Dr. Darcy Delenko, Nathan, Raksha, Stephen, Goodwin, Yong So Pa from Aju University, Dr. William Wang, and of course our lab PI, Dr. Tiffany Cruz, Dr. Carlos Gondora, and all the lab members. The funding, these projects were funded by the Indiana Core Marketing Council grant, as well as the Ag Initiative 2.0 grant. And I would also like to thank my sponsor for my PhD, Monsanto Bishop Borlaug, the International Scholars Program, allowing me to actually provide the data that I've shown you on the earlier slides of my presentation. Thank you very much, and I'm ready for you.
So we did have one question come through the chat, Daeyoung, and Damon asked, is there a substantial difference between the juvenile and the older leaves?
Substantial difference between the juvenile and older leaves in terms of.
Well, yeah, I guess I'm just wondering how predictive the very early symptoms are in terms of yield ultimately.
So the SCDA is relatively, if we just compare the two approaches between masked RCNN and SCDA definitely outperforms and is pretty reliable. But then I still want to improve it a little bit more in such a sense that I'm confident that the SCDA actually detects the stromata and not other artifacts. Because if we use image analysis, we're using with literally images. So we cannot try to rub off the thing that we think that is artifact or not. So yes,
Well, I guess I'm thinking more in terms of prediction of yield. Like if I'm a farmer and you say it has severity X in juvenile leaves, so I can tell you you're going to lose 30% or something, or is it much harder than that?
I think that's the direction that we really want to head towards too. Yeah.
Well, how good is it now?
To be honest, I can't give you a straightforward answer right now because that was not our priority at this moment. But then as we move forward, I can update you on that if we start working on predicting yield, depending on. Yes, thank you. Oh, I'm sorry. He has been waiting.
whole.
I noticed that the first signs of the disease peak around 70 some days after.
Mhm.
Immediate. But the infection, when the pathogen actually initiated infection, that happened
Much earlier.
much earlier.
Yes.
Do you have any idea how long does it take between infection and it starts, and when you start seeing the food bodies?
Right. So that was actually one of the projects that we started a couple of years ago. We're using hyperspectral imaging to see. We have been collecting data much before the disease symptom onset, and to see how much time interval does it need for incubation, infection, up to the visualization of symptoms. But through this technology, we can't know that.
Yeah.
I mean, we cannot address that question, but then we're trying to use other sensor imaging approaches as well, to kind of answer that question. Because I think that we're really, really interested in that question as well.
But so what do you think that it starts so late, like 78 days, it's already had plants have already pollinated and 3-4 weeks into grain section, right? Is it because the infection cannot take place much earlier? And that's something actually also raised to Damon's question as well. He's talking about juvenile and adult leaves, and that transition takes place from week 5 or week 6 of juvenile to adult leaves. So if there's any, anything about development as to do with.
Right. I think that's a really good idea, and that's also one of the questions that we would like to answer. But perhaps our RGB sensor is more geared towards to actually detecting what is visible through the human eyes, because we need the reference data in order to actually test the accuracy of the approach as well. So we're in the visible range of things right now. So the current standing we are here is, the goal is to detect tar spot stomata as accurately and as early as possible. Is the range of the project. That other question that you had is more of another project using other sensors. But that will be really.
So just to follow up on that, not necessarily associated with this project, but we have also been working with other projects where we're trying to develop a protocol that can be used to infect the plants and understand all these processes happening. But this is through experience, and this is why it's important to collaborate with people in other places, etc. So the reports that we have gotten is that the disease can be seen visually as early as V3 under some environments, some hybrids. I think here in the US, we have reported V5 around that and later. But unfortunately, we don't understand completely what that means in terms of the impact of yield. So that's why it's important to collect adequate information and then extrapolate and determine what is going on in terms of those infections and yield. But the scope of that Young's project was limited to developing that tool just now.
Perhaps using other sensors to try to see whether there's a difference in signal between healthy versus senescent versus diseased, and within the disease, whether it's a hemibiotropic cause disease, necrotrophic or biotrophic. I think that those questions are currently investigated using hyperspectral imaging as well. So yeah, I think that's a good avenue to start addressing this question. Thank you. Thank you for reading.
Right. So for the interesting presentation,
Thank you.
I will not slide on hyperspectral automated manufacturing. Anyway, so my question would be similar to what you and the image. What's the sensitivity the sensitivity itself, and also have you checked false positive rate
Yes.
in your time lapse experience? Like the black dot is a real symptom across all the time.
Right. Yes. All of you are providing us a lot of good questions, and also the questions that we really want to address. And we're starting that journey from using RGB sensors. Our main goal, actually, one of the main goals is to, aside from quantifying those leaf samples that are heavily infested with stromata, but also those that have like 1 or 2 stromata. And that's why we also utilize the method of image blocks. Because with image blocks, we can actually do a deep dive, a more zoom in assessment of that region. And that will allow us to allow the raters to actually be more sure about their assessments. And so in terms of sensitivity, we have tested the precision, recall, and dice coefficients. We have calculated those to test the amount of false positive, false negative, and the harmonious mean of those two. And we have observed at least more than 80%. And we have also tested the accuracy by stromata counts, stromata area, the location of the stromata. So basically, if a specific rater would say, this is a stromata, the algorithm should also detect this as a true stromata. And we have tested that as well. And we also observed good performances of that. With regards to how sensitive it will be, I think that's a really, really $1 million question, because to be honest, if the images are all variable, meaning if they're taken in all different ways, all different sensors, all different lightings, then this would not be a big size fits all. So it needs to probably be customized, but then that's why I have not closed this project after we publish. We're still working on that to improve that. So that when we have the smart app and when we allow the deployment of this tool in TARSpot research groups, then that would provide that the algorithm would provide more flexibility and sensitivity in quantifying those. And yeah, with regards to yield, I think yes, we would really want to work on that. Thank you for the question.
We have a couple questions from our Zoom audience. First one, have you tried other color systems such as HSI or lab, etc.?
HSVN, yes. I actually have tried those methods during my PhD as well. But then I think it's a matter of selecting one and pushing through. And I prefer this color space.
Next one, nice presentation. If I understand correctly, you scanned in the leaves, right? What is the potential for using this approach for quantifying disease on tissues that may have some curvature to them? Or perhaps using these types of algorithms to quantify disease severity from 3D images or point clouds?
So for the first question, you're right. We used a flatbed scanner, and that was based on the morphology of the sample that I collected. Those were rice leaves and had a more flexible type of tissue with less, actually nearly no wavy margins. Unlike corn, it's really a bit top, and it has its distinct wavy margins. So I wouldn't use flatbed scanners for that. One of the reasons why I also didn't use that is that it creates shadows when you press the tissue towards the scanner. And then that just makes another issue that we need to tackle. But then for tissues that are more flat, like rice leaves, I think also for leafy vegetables, which has flat surfaces, I think that would be flatbed scanners would be a good, affordable choice. As for your question with regards to point clouds, could you repeat that question once more with point clouds?
Or perhaps using these types of algorithms to quantify disease severity from 3D images or point clouds?
I think it's definitely possible. Yes, definitely possible. But then I haven't tried those yet.
I think we've got time for one more question. Darcy, if you are on and you'd like to unmute yourself and go ahead and ask.
Hi, Young. Hopefully you can hear me. I'm driving, so I apologize. That's the only way I can do this today. Anyhow, my question is, have you looked at comparing, so the tar spot stromata to those other diseases that we may be closely mimicking the symptomology? So I'm thinking like rust teleospores from southern or common rust or insect frass. So those are the two others that we're commonly getting confused, especially early on when we don't have a lot of tar spot out there, when trying to find those first lesions and detect the disease early. So have you
Right.
looked at comparing it to other diseases that are comparable with the symptomology?
Yes. To answer that question, yes, we have started that endeavor of trying to see whether SCDA can actually decipher stromata versus rust pustules plus versus not insect frass though, artifacts, and plus southern leaf blight symptoms as of this point. And I think that's important, and that's what we're trying to do right now. Just do one or two more diseases before we actually conglomerate that in the tar spot severity toolkit, wherein we can disseminate to different groups. I will keep you posted on that. I'm currently working on that.
Thank you.
Thank you.
All right, thank you very much, Dayon. Audience for joining us as well.
Thank you for coming. Thank you all, those who are in this.
Thank you. No problem.
Sorry about that.
So what happened?
Well, my computer crashed and then I was able to get back on. But I
Okay,
think the battery was too low.
and so the battery died.
Yes.
So was it recorded, the part that,
As soon as I logged back on, it was still recording.
oh, was it?
So I'll check the
Yeah,
recording.
check it because it's very
Yeah.
important. that we-
Yes, I know. I'm
It's
so sorry.
recorded. Yeah, all right.